Hello dear fans. What is cloning? Cloning in the broadest sense of this concept is a method of obtaining several organisms that are completely identical to each other through asexual reproduction. There are many living organisms in nature, the reproduction of which occurs in this way. Today, the term cloning is usually understood as obtaining copies of cells, genes, unicellular and even multicellular organisms by laboratory methods in an artificially created environment. In the Russian language, the term cloning came from the English klon, which, in turn, came from the Greek word for a twig, escape. This was the name of a group of plants that were obtained from one producer plant vegetatively, and not through seeds. These plants had exactly the same qualities as the plant from which they were obtained. Subsequently, each descendant plant began to be called clones, and their receipt was called cloning. With the development of science, the term began to be used with respect to cultivated cultures of bacteria, which also repeated the qualities of the producer organism, like plants, due to the genetic identity of all clones. The term cloning began to call the very biotechnology of obtaining identical organisms, which consisted in replacing the cell nucleus. The first experiments in cloning complex, multicellular organisms took place in the 50s of the 20th century. The object of their conduct was a frog. For this, they took a tadpole cell and transplanted it into an egg. Subsequently, a tadpole grew from such an egg an exact genetic copy of the original tadpole. Similar experiments were actively carried out in all countries of the world using various experimental objects, including mammals. In the course of the experiments, the embryo of the organism was isolated at the earliest stages of its development. Then the cells of the embryo were separated and placed in unfertilized eggs, from which the nuclei were removed. All cells of the embryo are characterized by the same set of genes, and the eggs acted as a kind of incubator for them. From these cells, embryos were grown, which were implanted into the uterus of females of this species, after which she gave birth to identical cubs. In 1997, not an embryo was cloned for the first time, but an adult mammal. The first such clone was the world-famous Dolly sheep. The author of this sensational experiment was a scientist from Scotland, Ian Wilmot. The lamb clone was obtained from the mammary gland cell of an adult sheep. For this, cells of this type were cultured in a medium containing a minimum of nutrients. Thus, the cells were not able to perform adult functions, differentiating to the state of the embryo. This cell was combined with the egg of another sheep, previously devoid of a nucleus, and the developing embryo was implanted into the uterus of a third adult female. The result is a full-fledged baby, with genetic material identical to the adult sheep from which the original cells were taken. After successful experiments with other mammals, in the late 90s of the 20th century, ideas began to appear to use the same technology for human cloning. This question has caused a storm of discussions in scientific and public circles. To date, most countries have signed the Convention on the Prohibition of Human Cloning. Thanks for watching.